Good afternoon, everyone. I'd like to welcome everyone to the mock peer review hosted by the Professional Development Core of the Dakota Cancer Collaborative on Translational Activity. My name is Christian Burish, and I'm the coordinator for the Professional Development Core for the University of South Dakota in Vermillion, South Dakota. I'll be your host for today's mock peer review. I arranged and planned today's activities, and I'll be here to watch the time and manage any technical difficulties.
Hi, everyone. My name is Miranda Rader. I am a project manager and coordinator for the Clinical Research, Resources and Facilities Core. I am from Stanford Research in Sioux Falls, South Dakota. I will be the Scientific Review Officer, or SRO. It's my role to arrange the logistics of the peer review, recruit and prepare reviewers, and ensure NIH regulations and procedures are followed during the peer review. Following the peer review, I'll assemble summary statements for each applicant.
Hey, I'm Don Sens. I'm going to be the chair of the study section. And I'm the co-director for the Professional Development Core. And I reside at the University of North Dakota in Grand Forks. And as a chair, my role will be to introduce applications and reviewers and guide the discussion to ensure it stays on topic. I will conclude and summarize each discussion and record the assigned reviewers and panel's final scores. Thank you.
Hi, everyone. My name is Tabitha Lemke, and I'm a project manager and coordinator for the Clinical Research, Resources and Facilities Core from Stanford Research in Sioux Falls. I will be the NIH program officer. As a program officer, I wrote the RFA for the applications to be reviewed today. The SRO allows the program officer to give a description of the RFA before the review and answer questions about the RFA that arise during the review. I will also observe and take notes on the discussion.
And now I will introduce all of the members of today's study section, beginning with Dr. Mark Williamson of UND. Are you out there today?
Yes, I am. I am Dr. Mark Williamson. I am the statistician and coordinator for the Biostatistics, Epidemiology, and Research Design Core at the University of North Dakota in Grand Forks. My primary background is general biology, though my current research is in epidemiology and statistical methods.
I understand that Dr. Sindel couldn't be with us today. Dr. Baugh, where are you?
Hi, I'm Dr. Lee Baugh. I'm an associate professor in the Sanford School of Medicine at the University of South Dakota. I'm also the other co-director for the Professional Development Core of the Dakota, and I am a cognitive neuroscientist by training.
Next up is Dr. Mel.
Hi there, I'm a Dakota scholar, and my area of expertise is I am a social behavioral epidemiologist.
Next up is Dr. Dagelberg.
Yes, hi, I'm Ashley Dagelberg, and I am with All True Health System. I manage the research department. I'm a physical therapist by trade.
Last but not least, Dr. Rowe.
Dr. Rowe, I believe you may be muted.
Sorry about that. Hi, everyone. My name is Sunyi Rowe. I am the professor for the Department of Social Work from the University of South Dakota in Sioux Falls, South Dakota. I am a Dakota scholar, and I will be the reviewer of improving HPV vaccination application. Nice to meet you all.
Right, now we'll go into the parameters of today's events. What we're about to witness is a mock study section involving two applications. These applications are from the National Cancer Institute as examples of successfully funded R21 applications. These applications only contain the specific aims and the research strategy. At this time, we'd like to give credit to the principal investigators, their respective institutions, and the National Cancer Institute for access to these applications today. The discussions are abbreviated in the interest of time and content. This means that the budget, biographical info, investigators, resources, environments, human subjects, invertebrate animals, and key authentication of biological and chemical resources are excluded because they're unavailable. Feel free to ask questions when we open the floor to Q&A. We'll have at least 3 opportunities for Q&A during today's events. If you're unable to ask a question during the time that we have, please reach out to me, christian.burrish@usd.edu, and I'll put my email in chat and we'll find the answer for you.
I'll summarize the scope and the sequence of events for each application that we'll review. The SRO asks the NIH program officer to summarize the request for applicants or RFA, and the NIH officer summarizes the RFA. The chair will take control of the meeting and introduce the applications and assign reviewers. The assigned reviewers give preliminary overall scores ranging from 1 to 9, with a score of 1 being the highest score possible and a score of 9 being the lowest score possible. Reviewer 1, also known as the reader, will give a brief overview of the application, with which 2 to 3 sentences describing the specific aims and the strengths and weaknesses of the application by the following criteria: significance, innovation, and approach. Then reviewer 2, also known as the primary reviewer, and reviewer 3, also known as a secondary reviewer, will describe the strengths and weaknesses of the application by the following criteria: significance, innovation, and approach. Reviewer 2 and reviewer 3 can add comments that they may have left out or disagree with the preceding reviewer. They may also occur with the preceding reviewers to avoid redundancy. After the assigned reviewers speak, the discussion opens to all reviewers. At this point, reviewers can ask questions to seek clarification from the 3 assigned reviewers and provide information on the research field that the application covers. As the discussion winds down, the chair will summarize the discussion based on core score driving criteria and noting areas of consensus and differences of opinion. The assigned reviewers will give their final impact scores and indicate the range that these scores occupy. For example, if the highest final score given by an assigned reviewer is a 2, and the lowest final score given by another assigned reviewer is a 5, then the range is between 2 and 5. The chair will ask if anyone on the panel will be scoring outside of the range. If so, the chair will ask the reviewers scoring outside of the range to raise their hands to indicate this, and the SRO and chair, so it can be noted on the summary statement, and that concludes the peer review process for a single application.
This is Don Sensback as the chair, and we'll be looking at the scoring criteria. And the significance is one of the big issues in scoring. And you'll evaluate whether the project addresses an important problem or critical barrier. What's the rigor of the prior research that serves as the key support for the proposed project? Does it extend and improve the knowledge and the technical capability in clinical practice if it's successful? And does it change the concept, methods, technology, treatment, services, preventative intervention in the field? Now, if you're doing a clinical trial, it's a little bit more involved. You'll have to, the scientific rationale and need for a clinical trial to test a proposed hypothesis or intervention is well supported by preliminary data or preclinical studies or information in the literature or knowledge of biological mechanisms. This is a hurdle that really has to be covered. For trials focusing on clinical or public health endpoints, is the clinical trial again necessary for testing and safety, efficacy or effectiveness of an intervention that could lead to a change in clinical practice, community behaviors, or healthcare policies? For trials focusing on mechanistic, behavioral, physiological, biochemical, or other biomedical endpoints, is the trial needed to advance the scientific understanding of the study? The other review criteria, it's very important as an investigator, is one of the important components will the productivity of that investigators in terms of impact on their work on the field, not by a number of publications or the impact factor, but how this actually impacts on the field of the grant being proposed. And is the PI the ability to lead the proposed project? If there's a collaboration, you need to support this by a letter or a history of previous collaboration. You really though would like to have a letter for collaborators in the grant describing their role and what they're going to do for you. And early stage investigators, new investigators, if you have the appropriate experience and training, they may not have a lot of publications and not required to have mentoring. Again, in this type of thing, collaborative letters do help. Established investigators, you're expected to have an ongoing record of significant accomplishments. Multiple PIs, these will require a separate heading, and you must have complementary and integrated expertise and whether the MPI plan is appropriate. This will be reviewed by the study section on whether or not the multiple PIs are actually needed. For clinical trials, do you have the ability to organize, manage, and implement the trial and meet the milestones and timelines? Have you appropriate experience in study coordination, data management, and statistics? Data management and statistics will be really looked at very carefully. For multi-center trials, have an appropriate organizational structure, and have you identified a core of potential center investigators for a coordinated center? These are really very essential for a clinical trial. Innovation. This is one of the hardest things to sometimes judge by a study section, and really does it employ novel theoretical concept, approaches, methodologies, instrumentation, or interventions to shift research or clinical practice. It can also propose refinement, improvement, and new applications of theoretical concept, approaches, methodologies, instrumentation, or intervention. It's one of the more hardest criteria for reviewers to evaluate. And you get a separate section to write a small thing on what is your innovation. Clinical trials, again, what are the innovative elements that enhance its sensitivity, potential for information, or potential advanced scientific knowledge or clinical practice? This is a very important scoring criteria. Approach, evaluate the project, presents, addresses. You know, you have to have a well-reasoned approach, good methodology, analysis. You have to have plans to address weaknesses in the rigor of prior research, have a sound and rigorous experimental strategy, definitely address potential problems, alternative strategies, and benchmarks. And if you don't have these in an application, it's not going to go anywhere. You have to identify your potential projects, your expected results, and alternative strategies. And sometimes this is a good place where you can put in an additional study if it's necessary, if one of the strategies would be an alternative strategy. Have adequate plans to address relevant biological variables, such as sex for studies in invertebrate animals or human subjects. Is a lot now studies require both male and female animals to be used in the study, and the same for human studies subjects. So clinical trials, again, justified and appropriate study design, adequate power for the goals of the study, uses an appropriate study population, size, gender, age, demographic groups, potential ethical issues. You have to identify these if there are any, and include plans to ensure robust data collection. Again, a really important requirement of a clinical study. Have appropriate planned analysis and statistical approach, and address methods for standardization of procedures for data management to assess the effect of interventions and quality control. These are really essential elements in a clinical trial. And if any of them are missing, you're going to have trouble under review. The environment is whether the scientific environment can support the project. Does it contribute to the project's success? Are there appropriate resources, facilities, and equipment for the project? And has unique features that will benefit the project. It's not dependent on the prestige of the institution, although sometimes this comes up, but most study section members will ignore the stature of the institution and go more with the environment. For clinical trials, the appropriate administrative, data coordinating, enrollment and laboratories testing center for the trial, and has the capacity and ability to conduct the trial at the ProSO sites and available enrollment centers. For international sites, application addresses the complexity of executing the clinical trial. For multi-site centers, again, has the ability of each site to enroll proposed numbers, adhere to protocols, collect and transmits data in a timely fashion, operate in the proposed organizational structure. These are areas where the DaCCoTA CTR really has a bunch of different cores to help you with this type of study. Okay, overall impact. That's your likelihood of for the project to exert a substantial, powerful influence on the research field. It's driven by the importance of the problem in the field and the study question being asked. Is overall impact can be influenced by all 5 scorable criteria: the significance, investigator, innovation, approach, and environment, and score driving criteria prioritized according to reviewer judgment. Is the impact score is not an average of the 5 criteria. So one criteria could be so bad it will influence the overall impact, or so good that it'll influence the overall impact. Comment on resubmissions and a renewal. It's really great if you get a score in your initial application, because usually when you resubmit or you're renewing, especially on resubmissions, the score usually gets much better if you can meet the reviewer's comments. So it gives the study section a guideline from the beginning score of what to go with. And on a triaged application, you don't get quite this effect. Again, consider the inclusion of women, minorities, age, cross lifespan, protection of human subjects. For a clinical trial for the overall impact, study the timeline described in detail, include startup activities, anticipated rate of enrollments, feasibility and justification of the timelines. In other words, you don't want to propose something that can't be done within the time that you're going to be funded. Incorporation of efficiencies and utilization of existing resources, potential challenges and solution, human subjects, the appropriateness of the DSMP or the DSMB. Human subjects will be looked at really closely for a clinical trial and its impact. So your overall impact score is, if they're high, is usually between the score of 1, 2, and 3. And pretty much you're addressing a problem that the study section is deemed to be of high importance or interest to the field. You may have some or no weaknesses. A score of 1, if you're ever on a study section, be ready to defend that score because you should have really brought up no weaknesses. And at 2 or 3, you can have some minor weaknesses, and usually they'll be stated as not having much of an impact on the ability to do the project. 4, 5, and 6, again, you could have addressing a problem of high importance in the field. You could have weaknesses in the criteria that bring down the overall impact to medium. So your importance could be high, and maybe your environment is not so good, so your score could come down. And it may be addressing a problem of moderate importance in the field and not have any weaknesses actually in the operational part of the grant or the approach. So if you get up to 7, 8, and 9, pretty much here you're addressing a problem of moderate to high importance, but a lot of weaknesses in the criteria bring down the overall impact very low. And again, you could be addressing a problem that has low or no importance in the field and actually have not that many weaknesses in the project, but just have a weakness in why it would even want to be done. So that's the score criteria for your impact. So the individual scores, if you have a 1, it's exceptional, and you find no weaknesses in that application. What you'll find in some study sections is the person will give it a 1 and have a bunch of weaknesses, and the chair will intervene, or some of the reviewers will intervene to say, you can't do that if there's weaknesses. And outstanding, extremely strong, negligible weaknesses. And the weaknesses really won't impact on the ability to complete the study. It would be minor weaknesses that might improve the study overall. Excellent, again, very strong with only minor weaknesses. Very good, strong, but with numerous minor weaknesses. Good, again, strong, but with at least 1 moderate weakness. And if you get 6, 7, 8, and 9, you've had some strengths in an application, but there's going to be a lot of moderate or major weaknesses in the overall thrust of that application.
All right, we have just a few minutes for questions before we go into our first review of the day. So any questions? Let's hear them.
We need to cover a few legal issues before we begin this peer review. Everything discussed in this room must stay in this room. Don't go discussing this review with your colleagues, collaborators, co-investigators, or even someone across the hall from your home institution. If you have a conflict of interest, it must be identified ahead of time. If anyone here has a conflict of interest with any applications being discussed today, we'll have that person leave the room for the duration of that discussion. We also ask that you don't discuss applications previously discussed or applications yet to be discussed, because others may be in conflict with those applications. If you suspect scientific misconduct on the part of an applicant, the reviewer should privately contact the SRO, preferably before the review commences. We emphasize that the applicants get the benefit of the doubt in accusations of scientific misconduct. I'd like to ask the NIH program officer to summarize the RFA, since she was involved in writing the RFA.
Today, we'll be reviewing the applications from the RFA from NCI National Cancer Institute named Dissemination and Implementation Research in Health. The objective of the RFA is to identify, develop, and refine effective and efficient methods, structures, and strategies to disseminate and implement research, tested health behavior change interventions, and evidence-based prevention, early detection, diagnostic, treatment, and quality of life improvement services into public health and clinical practice settings.
Okay, I think I'm up. So we've heard from our, no, we got to get to our assigned reviewers. So did we summarize the RFA yet?
Yes, we summarized the RFA. So the chair takes command of the conversation.
Okay, so this is now, so the RFA has been summarized. And so now when presenting your evaluation, please focus your discussion on the score driving issues, which are significance, innovation, and approach. And if you're anybody in conflict that any of these people, or the schools, or you have any relationship with the person who submitted the grant, we'll need to remove you from the review. So our first application is from Brian Weiner from the University of North Carolina, Chapel Hill. Title of the application is Increasing Colorectal Cancer Screening Rates in Community Health Centers. The assigned reviewers are Mark Williamson, Sandeep Singhal, which is going to be missing, and so we'll only go with 2, and Leebaugh. So if you would please give your primary impact scores.
Okay, mine is a 4.
Okay, Lee?
I'll go with a 3.
4 and a 3. Okay, so what I like is the primary reviewer to give a short summary of the application, just a few lines, and then move on into his strengths and weaknesses of the application by the assigned criteria, significant innovation and approach.
All right. So this proposal aims to use a colorectal toolkit with 3 pieces and an outreach specialist to improve colorectal cancer screening rates in community health centers, with 3 aims of: 1, assessing the extent of implementation, that is, what's already being done there to improve screening. 2, estimate the cost of implementation and maintenance, that is, how hard it is to change. And 3, conduct a pilot study on the changes implemented. Ergo, how well it actually works to implement the toolkit and outreach specialist. So what I found in the significance section for strengths, they are trying to improve screening rates for underserved and high-risk groups. So that's very important, especially in the community health center arena. And because the community health centers struggle with getting enough screening recommendations, there are definitely methods that are needed to make this doable in the environment. And so they're really trying to do just that. Now, a weakness I found in the significance was that increases in colorectal screening success may not be meaningfully higher. They cite some previous work, which saw between a 4 and 13% yearly increase, but that was in a bigger system with more intensive collaborative model. So I could see if 4% would be at the low end for a more intense model, a less intense model they're trying to implement here could be even lower. So it might be negligible increase in screening rates. On the innovation section, I found some strengths. I really liked how they looked at a very realistic way to improve screening that doesn't require massive resources. Community health centers often are overworked and understaffed, so simply trying to do more with less isn't a good solution. And some of their attempts haven't been applied to community health centers specifically. And they also actually focus on implementation cost to see how this would run in the long term. Now, a weakness I found in this is some of this sounded more like optimization rather than innovation, kind of a middle of the road version between what's already done elsewhere. They talk about a sort of low intensity train the trainer model used in some studies for community health centers. And then the more resource heavy collaborative model in more advanced or bigger areas. And so this sort of sometimes seemed like just between the two of those. Then finally, in the approach, some strengths I found, they really did a good job of outlining their technical and staff support. This seemed to be really good approaches to actually helping the community health center with tangible support. And it really identified the 3 components of the office system. That is the toolkit they mentioned. They have a solid plan for timing, scheduling out what they're going to do when, and well-researched and competent schema for getting specific community health centers included in the study and retained. So they did a lot of research on the right targets. One of the weaknesses I found was the actual day-to-day uses of some of the toolkit items. Some of the examples of the toolkit being things like sample policies or tracking logs. It was kind of vague on how they would actually be used. It might be some of my knowledge weakness of these sort of systems, but it wasn't always clear how that would work. And then I wasn't sure so much how the prior research was more than just talking with some of these community health centers about their needs and conducting some research, some focus groups. And then the final weakness I found was some of the working hypotheses on the implementation feasibility seemed rather weak on falsifiability. For example, they had, one of their working hypotheses was, with outreach support, community health centers can implement 3 tools and create implementation climates that promote consistent colorectal cancer screening recommendations. I could see that that's very easy. There's not a clear guideline of like, well, what does success look like? What does failure look like? So it was a little weak there. And with that, that is my overview of those 3 areas.
Oh, thank you very much. Now we'll go to Dr. Ball and Lee. Would you give us a secondary review of this, please?
Absolutely. So I'd like to start by thanking Dr. Williamson for the wonderfully concise review. It'll make my job a whole lot easier. So I just have a couple of things to add. I ranked innovation slightly lower than I typically would, as this approach, as was mentioned, really seems to be a combination of previous techniques that proved to have mixed success. And I wasn't entirely convinced that what they're doing here is going to be a huge step forward in the field. Similar to what was pointed out before, the preliminary studies consisted primarily of focus groups, and I think the application would be strengthened by preliminary data showing the potential of the CRC screening methods being utilized rather than just asking focus groups what they think about this technique. Now, with this being an R21, I didn't have that affect my score too much because although they can include preliminary data, it's certainly not needed for an R21 grant. I thought that the project would benefit from having a statistician as part of the project team to consult on a more sophisticated statistical analysis. The analysis that was presented, while competent and I didn't see any errors with it, I think they could do a lot more with the data that they're collecting. And then finally, I thought it might be a bit of an ambitious timeline for IRB review and approval. I think they had about a month in there to accomplish all of that. And with the complexity of this project, I think they might have been a little bit hopeful in being able to complete it in that amount of time. But those were my additional comments. Thank you.
Thank you very much. So now what we can do, we've had the primary and secondary reviews. Now we can open the floor to the rest of the assigned review panel and see what kind of questions they might have for the reviewers. So this is the chair. I do have a question. And so what were they using for screening? Were they going with something like a Cologuard, or were they actually, still going to use, the regular scoping procedure, or how were they going to do this? Or was it more just to get people to get screened?
Yeah, it was more focused on the getting people to get screened
That's what I
and
thought.
in improving the rate of people who were eligible for the screening to actually get asked and reminded to. I think that was definitely one of the limiting factors. They do talk a little bit about screening methods. I'm trying to go through here. I know they definitely brought up things like fecal blood tests as a potential thing to focus on especially if there's limitations in funding or affordability for more rigorous like colonoscopies or things like that.
So it's pretty much what targets a lot of the underserved communities that don't have the clinics and things right there to get people to get this done.
Mm-hmm.
That's really important. Any questions from anybody else, please? You got two experienced reviewers. You can get some information from here.
So perhaps I have a question that did the group try to consider also other chronic disease associated with colorectal cancer, such as obesity or diabetic disease? Did they they highlighted that this is relevance to this screen because obesity is a direct impact on the colorectal cancer? Or they missed to mention about that?
So I can speak a little bit to that. That from my recollection of the grant, they did not mention those. Their primary focus is on leaving or looking at the ability to have people transition from the, hey, you should get a screening to actually getting that screening done. Therefore, they leave all of the clinical considerations to the physician in this case. So if the physician feels like they should have a certain type of screening done, or there are certain factors that should indicate that they should get a screening versus they shouldn't, that's not something that this group was concerned with. They want to see if their technique or their approach increases, if you think of it sort of like a sales pitch, you know, from making the pitch to actually having the person buy the product. In this case, making the pitch, hey, you should get a screening done, to them actually going on and getting it. But that is a good point, Dr. Resvani.
Thank you.
Okay, any other questions from the panel? And if not, then what I will ask is that each member, each of the reviewers restate their scores for the application after hearing each other's reviews and the questions from the panel. Mark.
Yes, so I'm going to stay with a 4.
Okay.
And after
Lee.
hearing that discussion, I'll move to a 4 as well.
Okay, so we have 2 4s, so there's no way to, so the range is 4. So if you're going to go and score anything but a 4, you need to raise your hand and let us know. And for the most part, the reason we do this is if you go outside the range, we may want some extra comments on why you would think that this was either better or worse than what you've heard. So these are very valuable to the applicant in the study section summary that the applicant will get. So if you're going out of range, it's really important that not so much the chair needs to know it, but the SRO needs to know it too. So, and I think after that, I'll summarize this real quick. I think I was already supposed to do that, but I'll do it anyway now, a little late. But so summarizing this, the strengths of this was it's a substantial problem to try to get underserved and other communities to get screened. So that's an important part of the strengths of the application. And what you want to do is improve the screening rates. This is a bit of a new methodology is a strength here that they're proposing to try to do this. They had a pretty good timeline. It seems feasible. So, you know, it was a well-developed study. It has some minor weaknesses, maybe even a moderate weakness. Is one of the things that really impacted here was there was a weakness in their preliminary studies. This looked like they only had focus groups to say this really needs to be done. And it would have been nice if they could have had a little bit more of this. And on the expectation of what your expected results are, is what is success? They really needed to define what the success markers were going to be for this project. And pretty much there's no new techniques here. It's just optimizing what's already there. And so these minor weaknesses sort of dropped it into the into the score of 4, which I believe is good or very good range, where you have an important problem, but you have some significant minor weaknesses or maybe a moderate weakness in the experimental design of the project. And if you're really on a study section, I would have done that before I got final scores.
So now is a good time to have some Q&A to go over the first application that we just reviewed today, anything on scoring, anything on procedure. We have time for a few questions from today's audience at this time.
Yeah, the one thing as chair I'd just like to comment on is that what the NCI does is they have an omnibus study section for all R21s, which is nice because you get a percentile score. On some RFAs, if it's not a regular standing study section or an omnibus, you'll get your score, but you won't get a percentile. Okay, because they don't have anything to compare it to. So NCI sort of does this in a nice way where they have enough data to actually rank the grants on a percentile basis. A lot of RFAs won't. You'll just get your score.
If we don't have any questions from the audience at this time, I'd like to go into application number 2. The chair has the floor.
Okay, so our second and final application is from Benjamin Teeter from the University of Arkansas for Medical Science. The title of the application is Improving HPV Vaccination Using Implementation Strategies in Community Pharmacies. Signed reviewers are Soon Hee, Melanie Nadeau, and Ashley. And if you would please give your preliminary scores, Dr. Rowe.
3.
3?
Yep.
Okay. Dr. Nadeau.
5.
5? Okay. I just got to catch up with you. 3 and 5. And where's my people? And Ashley.
4.
4? Okay. So, okay, Dr. Rowe, you'll give the preliminary summary. Okay, brief summary plus your strengths and weaknesses.
Sure. This study has 3 aims. Aim 1, identify barriers and facilitators to community pharmacies' provision of HPV vaccine through a mixed method design with pharmacy staff members and local physicians. Aim number 2, select a pharmacist physician collaborative model and identify implementation strategies through an evidence-based quality improvement process with key stakeholders. And lastly, aim 3, pilot the selected pharmacist and physician collaborative model and implementation strategies in 2 pharmacies, 1 rural pharmacy and 1 urban pharmacy, and evaluate on relevant implementation outcomes. The long-term goal of this project is to improve HPV vaccination rates among adolescents in the US, especially in rural and underserved areas in Arkansas. While this study addresses significant public health issues, it has some weakness to address. I will go one by one. In section significant section, I found 3 strengths. Strength 1, this study addresses the importance of local pharmacy access to vaccinations for children provider I will use abbreviation VFC. VFC is a program administered by the Centers for Disease Control and Prevention. That provides free vaccines, including HPV, to providers for administration to children at no charge. Strength 2, this project collaborates with community pharmacies. Pharmacy participation in VFC can help increase HPV immunization rates. Especially community pharmacies are highly accessible when compared to traditional vaccination sites due to their extended business hours, no copays, and no requirement to schedule an appointment. Strength 3, this study use implementation science to determine and pilot test a promising vaccine delivery partnership model between pharmacists and physicians and an array of implementation strategies to support uptake and sustainability of HPV vaccine administration in the community pharmacy. I found one weakness in significance section. There is a lack of description of existing intervention to improve HPV immunization rates in an underserved area. I would like to talk about innovation section. I found 4 strengths. This study utilizes community pharmacies as HPV vaccination sites. Community pharmacies are established vaccination providers, yet very few provide vaccinations for children and HPV vaccines. Secondly, this project explores pharmacist-physician collaboration models. Pharmacists are certified to provide vaccines with appropriate agreement in place with physicians. However, little research has investigated the multitude of options available to establish effective collaborations or which are favored by participating parties. Strength is 3. This research incorporates key stakeholders to develop implementation strategies, such as pharmacists, physicians, patients, and leaders from HPV-focused community work groups will provide feedback on proposed implementation strategies to select and develop novel ways to implement HPV and VFC vaccines in community pharmacies. Strength is 4. This study use of research methodology that is grounded in rigorous research, existing literature, and also a mixed method they apply, and healthcare experience expertise on HPV immunization in high burden settings. I found one weakness. Researchers' use of community pharmacies as HPV vaccination site is not innovative. Currently, many community pharmacies are available to provide vaccines as free. And lastly, in approach section, I found 3 strengths. Strength is 1, researchers address clear transition from previous work. Researchers identify barriers and facilitators to community pharmacies' provision of HPV vaccine through a mixed method design with pharmacy staff members and local physicians. This is the novel of this approach. Secondly, use of evidence-based quality improvement to guide the assessment, such as research consensus on key barriers, facilitators to providing HPV vaccine in community pharmacies. Secondly, select a collaborative model to pilot test and develop implementation strategies to pilot study. Last strength is the researchers have a strong partnership with the community pharmacies. This is a surplus point. And I found 4 weaknesses. Weakness 1, feasibility is a concern because they are mentioned about each period, they are looking for at least 597 eligible participants and 3 points. So participant ranges will be 597 to 1791. This is a very large number, and I see that this is very ambitious. Secondly, this study needs a more detailed retention strategies for follow-up, 3 months time period. And sustainability of the intervention is not clearly suggested in the proposal. Lastly, they are not adherence to HPV testing, which means 5-year recommendation is not planned in the study. This is my summary.
Oh, thank you very much. Oh, Melanie.
Yes, thank you, Dr. O, for such a great overview of the strengths and limitations and summary of the proposal. So, and I agree with everything that you have stated. The thing that really stood out for me is that I guess there's this overarching assumption that access is the issue. So they state that requiring initially when this the vaccine first rolled out, there was 3 shots that were necessary, and then now there's only 2, and they thought that it would increase, and it hadn't. So it made me wonder if access is the issue. And so I'm concerned with the carrying out this what is being proposed here. If access is not the issue, then what? And then of course the timeline, the feasibility of what's being proposed. The strength is that it is a pilot, given it is an access issue, and innovative ways of identifying a strong implementation method to increase vaccination rates. The mixed methods approach is also a strength in interviewing key stakeholders and looking for there's many collaborative models that have been that show promise, and their method aligns nicely with identifying the best model given the situation.
Okay, thank you. Ashley.
Yeah, so I did find that the problem was of high importance with the highest incidence rate of HPV related cancers in Arkansas, which, you know, with implementation of the vaccine could be preventable. I did think there were challenges in that only 4% of physician providers were part of that VFC network, the Vaccines for Children, and virtually no pharmacists were. So it seemed like that was a caveat with being part of this study. And then, you know, we're looking at our population pool that has to be individuals under the age of 18 that have Medicaid or are uninsured. So it's not all children under the age of 18. So I think those are 2 issues that you have to look at. It decreases the sample size a little bit. And then what's the education in this second round? I know it's a pilot, but to providers and pharmacies to become part of this VFC network, you know, by the CDC. The challenges I thought for the collaboration models were The first option, it was tough because it was still a two-step process for the patient. They would need to go into the provider first and then get a script to go into the pharmacist to get their second vaccine. One of the other collaboration models was you can do both at the pharmacy, but they would have to have this protocol accepted by multiple individual providers. So that's a lot of legwork. And then the third, to me, was not a viable option is pharmacies. Yes, they have access, they're open weekends and later hours, but there is not going to be a time where probably a pharmacist can go in and do a vaccine clinic at a provider's office because they probably still need to manage the pharmacy. So I did see some, you know, a little bit of the approach with the collaboration models, I think will still be tough. There wasn't a lot in here written about the education component to the community about prevention and vaccines. So we know we have lower percentage rates for the vaccines in this community for both female and male. I would like to see data that compares it to other two-step vaccines. Now, I know there was data that said how much the pharmacies gave in terms of the flu vaccine and other vaccines, but it didn't say what the community rate was of getting those vaccines. So I thought there were some limitations with the data to begin with.
Okay, thank you very much. And what I can do now is I'll summarize the strengths and weaknesses of this. This was an interesting proposal that addresses a problem of trying to get HPV vaccinations rates to increase. It's a very effective vaccine. It's got a nice history of safety. And so the goal really is overall to improve this HPV vaccination rate. And they're trying to do this with a model of local pharmacies, which is a good place to get vaccinations. It's a little more, it's easier than going to a doctor's office or a clinic, which is another strength. So it's very easy to get the vaccine under this. And the innovation of this is they're trying to involve the key stakeholders that would benefit from this. There were quite a few minor to moderate weaknesses. And, you know, there isn't always a community pharmacy in a lot of underserved areas, which I thought was something that needed to be addressed. And maybe it was, and we just didn't bring it up. And this whole thing of using pharmacies for vaccinations really not very new and innovative. So the feasibility, they wanted a very large number of individuals to get involved in this. And there was some question on whether or not they could even find that kind of a high number. And, you know, I don't get the educational part of this. You know, the problem is that, you know, people don't always trust getting vaccines, and you've got to get through the level of trust here. And I didn't see it addressed. Maybe it was, but it seemed to me to be a pretty serious weakness if they weren't going to try to educate that this vaccine is safe and for everybody. And, you know, it's been tested extensively on like a lot of other things. So I just thought that, you know, from the reviewers' comments, it lacks some really important details that might lower the enthusiasm for the application. So we can open this up for the panel. If there's any questions, you know, I summarized it before the panel, but that's okay. They didn't have questions the last time. I have some. So we're open for questions from anybody to the reviewers.
Okay. Yeah, one question I had about this study, especially I think it's AIM-1, they talked about their mixed methods. They said they're going to be primarily qualitative with a quantitative assessment for what they call concurrent triangulation. Now, I'm not terribly familiar with qualitative methods. I mostly deal with quantitative. Do others think that this setup is sufficient sort of to capture the detail they want from these surveys? Focusing primarily on the qualitative side of it.
I'll just comment and say that from my assessment of this, they're using the process evaluation as part of their mixed method process. So the qualitative assessment and quality improvement of their process is part of that. And for what they're trying to do, along with the interviews. And so with those two things in combination, what they're trying to do, it seems appropriate.
Okay, great. Thank you.
As a chair, I have a question on the innovation of this. Is it really a very new approach? Because, you know, trying to get people to get HPV vaccinations has been, you know, around now for quite a long time. So is there something really innovatively new in this?
I mean, I think from my opinion, it's just having the HPV vaccine at a pharmacy versus in the provider clinic is the innovative part of it, you know. Yeah. That's the innovation, is just having that access in a different place as opposed to having to go in and make an appointment with your provider or pediatrician.
Yeah. So I guess one of my questions is, you know, I don't always know these things, is I've been getting all kinds of vaccinations at a pharmacy. Is having to get one for HPV more difficult because of the age of the person getting the vaccine?
Well, it's
I
stated
mean, I've
in
gotten flu, COVID, Hep A, and all kinds of things.
Well, it's stated in there that, you know, in order for a pharmacist to be able to give a vaccine, it has to be under physician oversight. I'm assuming that with the flu, there were protocols, a protocol that was already approved loosely with a provider. And I'm thinking because of this patient demographic, maybe it's a little bit stricter parameters. I'm not 100% sure versus the flu, but this being pediatric specific. Yeah. And then we're looking at, you know, socioeconomic status, you know, for somebody that's on Medicaid or, you know, maybe from an underserved rural patient population, there's just a little more oversight. But I just didn't realize, yeah, that pharmacists can't just go give vaccines without having the oversight from the physician. So that was.
Yeah, I didn't either. That was sort of my question. Yeah.
Yep.
Thank you. Any other questions? If not, you know, I'll ask the reviewers to summarize their scores.
I changed my score 3 to 2.
To a 2?
Yeah.
Okay. Melanie, you're
I'll stay at 5.
okay. Ashley.
I'll stay at 4.
Okay. All right. So that's the final scores. And what I need to know is anybody going to score out of range? And since nobody has, I am going to. And I'm going to give this a 6. And I'll have to write an explanation to myself on why I've done that in the summary statement. I just think that this has been around so long, I just don't see what's really new here. And I think the emphasis has to be on underserved areas. And I mean, most of the underserved areas I'm familiar with don't have a pharmacy, or a lot of them don't. And of course, I may be biased because of North Dakota and what I've seen, but I don't get this. So I'm out of range and I have to write something to myself, which would be about what I said. So now you've had an out of range.
And Following the end of that second review, Dr. Ball would like to have a word with our audience about NIH's Early Career Reviewer Program, or the ECR.
All right, well, thank you all for taking time out of your day to see our mock study section panel. Hopefully you found it interesting. I wanted to bring to everyone's attention the NIH's Early Career Reviewer Program. So this is a program that I was actually able to participate in what seems like many, many years ago, where you are given the opportunity to actually serve on a study section. So alongside many of the most accomplished researchers in your field, you submit a review, you go through the whole process that we just went through to help NIH identify the most promising grant applications. Through this, you get to see a lot of the details as to how reviewers determine overall impact scores. You get to sort of look at your own grant writing skills and compare them to the grants that you're seeing coming in, getting an insider's view of how grant applications are evaluated. You know, my first time being on an NIH study section, you know, I had always heard when people would say that, you know, formatting is such a big deal. It has to look like every other grant in order for it to score well. And you think, well, that's ridiculous. But no, sure enough, one of the grants came in, they just used a different font. It was technically an allowed font, but it was a different font. And that came up in the discussion and people were like, I'm not sure how much experience they have writing grants because they just chose the font that isn't typically used. Of course, you get to serve the scientific community by participating in an NIH peer review process. So you get to put that on your CV, which looks great as you're making that transition from assistant professor to associate professor. And you also could get to develop your research evaluation and critique writing skills. So I'll put in the chat box there the link for looking more into this. It's open to all early investigators. NIH defines that as people that don't have specific grants at the level of a PI, like an R01 or something. All of the exclusionary details are there. It also means that you have to be at the assistant level. You can't be an associate, even if you've never had an NIH grant. If you are an associate professor, you're not eligible for this program. And you have to have an active research program. So by that, you know, running your lab, being on papers, you need at least one paper as senior author, which NIH defines as first, last, or corresponding positions on the paper. But if you meet those criteria, definitely look into it. Like I said, I did this many years ago and really enjoyed the experience. You get to meet all of the people that trash your grants and put a face to it, and then it doesn't, you know, sometimes it hurts a little bit more when you know who it is, but other times, you know, perhaps not as much. And really, it's just a great way to see how things are done, and it allows you to craft your grants in a much more effective manner. Get your name out there to the people. So that's probably the much bigger side of things. So then when they're trashing your grant, they actually have a face to go along with it, and they know, oh, you know, this person made some really great comments on that study section, you know, last time around. Maybe they're not as much of an idiot as their grant makes them seem to be. We should give them the benefit of the doubt. Those sorts of things, I think, come up. So like I said, I'll put that in the chat. And if people have questions about that, feel free to send myself and Christian an email and we can forward along additional details. But like I said, did it many years ago, and I've served on many study sections since then. And it really was what got my foot in the door in a lot of ways for working with NIH in that manner.
Thank you, Dr. Ball. And for our last activity of the day, our last Q&A session. So any questions you've had about anything you've seen, starting from the latest with the ECR to the earliest we were talking about scoring, procedure, and roles, to anything that you saw in our review sessions, now's the time. Well, if no one has any questions for our reviewers, I'll go ahead and conclude today's activities. I'd like to thank everyone who participated in the panel for their time to prepare. This took place over many months. I think we were first talking about this in October, and here we are putting finishing touches on it here in March. I'd like to go ahead and thank our audience for taking the time out of their busy schedules to be with us and to witness science in action. Have a good rest of your day.
Okay, I just want to thank Christian for this, and he is on top of the professional development core like nobody I've ever seen. He will, if you send him an email, make sure it gets answered. He does a great job on this. So if there's no questions you one day ask here, send him an email and he'll get them out. Thanks, Christian.
Thank you, Dr. Sens.
Thank you, Christian. Bye-bye.
Yeah, thanks, Dr. Bro.
Bye now, everybody.
Farewell.